Hello friends, I'm Dr. Sutar from Sanjini College of Pharmaceutical Education and Research, Department of Pharmacognosy. So friend, today we are going to study a subject, Pharmacognosy and Phytochemistry second, uh, third year, fifth semester. In this subject, we are going to study a general introduction about the glycosides. So friends, you know that what are glycosides? Glycosides are nothing but the secondary metabolites are there, which on hydrolysis gives you two components. One is a sugar part, another one is a non-sugar part. You know what is sugar part? Sugar part is known as a glycon part. Sorry, sugar part is known as a glycon part and non-sugar part is known as a glycon part. So a glycon part is a steroidal moiety. So that is mostly responsible for the pharmacological actions such as the purgative is there, laxative is there, analgesic is there. Likewise, the different type of the actions are there, cardiotonic is there. So likewise, these are the different actions are there which is occur because of the glycon part. Now, what is the role of the glycon part? So glycon part is acting as a carrier, so which carries this glycon moiety to the particular site to show the particular pharmacological action at the site of the action. So this is the role of the glycon part. We can say that the sugar part, number of the sugar molecules are more, that is the it enhances the solubility of the component. The number of the OH group is also responsible for that one. So what are cardiac glycosides? Cardiac glycosides, or they are also known as a steroidal glycosides. Cardiac glycosides are of two types. One is a cardenolide and another one is a bufadinolide. So what are these one cardenolide and bufadinolides? So cardenolides is having a five-member lactone ring. So here you have observed that one. The five-member lactone ring is there that is present, which is attached to the C17 position to the this one steroidal moiety. So this is a cardenolide. What is bufadinolide? Bufadinolide is nothing but steroidal moiety is there where to which at beta 17 position the lactone ring is there is attached. So what is the difference between the cardenolide and bufadinolide? Cardenolide is a C23 steroidal moiety, whereas it have a five-member lactone ring. Here you observe in case of the structure, there is a five-member lactone ring is present, okay, which is attached to the C17 position, okay, and this one is there 23 number of the carbon atoms are there that are to be present. The third one point is there, it contains one double bond. At 20 number, it shows the double bond in case of the cardenolide. So, and the attachment through the C17 position is there, means this lactone moiety is attached to the steroidal moiety at the C17 position. Now, let's see about the bufadinolides. So it has C24 steroidal moiety, where a six-member lactone ring is there to be present. It shows two number of the double bonds are there that are to be present in case of this one structure. So, and this is lactone moiety is attached to the steroidal moiety through beta 17 position. See 17 position. Okay. So likewise, this is the differentiation points are there between them. So the sugar part is C3 beta linkage is there. Means in case of this one glycoside, that three position the sugar groups are attached, and the maximum number of the three sugar groups are attached to this one particular steroidal moiety. So what type of the sugars are there that should be attached? So glucose is known as a glucoside, glucose, glucoside, rhamnose, rhamnoside, digitoxose, digitallose, trimerose, sarmentose, and thiotose. Likewise, these are the sugar moieties are attached to the steroidal moiety. You know, at a time, three number of the sugars are there that should be attached to this one moiety. So why the name is so given? Bufadinolide. Bufalin is a foreign word is there, or we can say that Greek word is there. Bufalin that is to be obtained from the skin of the toad is there. So a poisonous one is there, and that poison we can use for the treatment of the heart also. Okay. Now let's see about this one is the lactone ring. Where is the lactone ring? Yes. In case of the cardenolide, this is the lactone ring is there. And in case of the bufadinolide, this is the lactone ring is there. Okay. Now, here the cardenolide and bufadinolide, which family shows the presence of the cardenolide bufadinolide? So, the Leguminosae family is there, Sterculiaceae family is there, then Scrofulariaceae family, Cruciferae, Ephorbiaceae, these are the families are there. Mostly the cardenolides are there that she found in case of the plant kingdom, whereas the bufadinolides are there that shows that is the Labeti, or we can see that Liliaceae family and Ranunculus family shows the presence of this one bufadinolide. We show the action on the heart. So friends, now let's see how the drug shows its action. Normally, when the heart there is not to be pumping properly, so the blood circulation to the particular site or the target organ is there is not to be sufficient as the demand is high. So in case of this one, the whenever there is a exchange, or we can say that the sodium as well as the calcium that should be present outside of the cell, and the potassium if there is inside the cell membrane. So there is a membrane depolarization is occur. That is an exchange of this one sodium potassium if the issue occurs. So for that one purpose, exchange ATPase pump is going to be provide energy. Sodium potassium ATPase pump that we are called. So the potassium which is present inside the sodium is going to be entered inside the cell of the myocardium, whereas the calcium is also to be follow the sodium, and in third figure the potassium is goes outside, the sodium as well as the calcium that remain inside the cell. Okay, so because of which the when we are going to take the digitalis, this digitalis is attack on this one sodium potassium ATPase pump, and it blocks the action of the sodium potassium ATPase pump at this level, because of which the sodium as well as the calcium is remain in higher concentration in the cell of the myocardium, because of which calcium triggers the intracellular action, means potentiate the action of the myocardium, because of which there is a forceful contraction of the myocardium, or we can say that the heart muscles are there, and in this way there is a complete emptying of the heart is occur. So in this way, the cardiac glycoside show their action on the heart. So in this way, the cardiac glycoside is there is to be used in the different type of the heart problems which are related with the force of the contraction only. Okay. In the next slide or in the next video, we see the details about the other drug. Okay. Thank you.